much, Malgré. First of all, for organizing and the flexibility to rearrange my lecture today. I apologize for the inconvenience. It was little I could do. But anyway, we were actually quite happy with this bilateral funding. Malgré visited us a few months ago, and now it was my turn to come here. It's actually very interesting to see how at different places science is organized, and we can also exchange perhaps ideas and potential common interest. Today, I will try to start by giving you a broad overview of what we have been doing during many years now, but mainly focusing on the part related to the bio applications that we have been doing for the last 5 or 6 years. So, before starting with the science, if this works. Okay. All right. So I wanted to show you where we are, where we work. So the place is the city of San Sebastian, which is within the region of the Basque Country, very close to the border with France, only 15 minutes by car. And one of the cities which is attracting a lot of tourism, and it is made of some of the locals, but you'll see why. Actually, the weather is not always like this, but in summer, usually, if you're not very unlucky, it looks like this. And this is where I usually swim in the evening after work. And so actually, as Malgré mentioned, I have a second lab at the University of Vigo, which is right here, very close to the border with Portugal, and with a landscape that looks very much like this. So I have another beach there to swim without there. So anyway, I found on the internet that the Germans called San Sebastian the happiest city in the world. And there are several reasons for that, that also Malgré can testify. I'm also trying to attract you to this, and I will tell you why at the end of the lecture. But, well, anyway, so this all looks very nice, but as I mentioned, before moving to San Sebastian, I was in Vigo, where I could also have the beach, very good seafood, and also waves. So this was not the reason that I moved there. The reason was that if you look at this other picture of San Sebastian from the inland, you see, first of all, the technology park, from where actually I had an office, now not anymore, but I had an office from which I could see the sea. But it is located in a very busy scientific and technological area, thanks to the large investment by the Basque government in the last 25 years or so. And so at Silvio Magune, this is a very funny name for an institute. Nobody understands why it is called like that. And it is related to the Basque region having an ancient language, which is the Basque language. They claim it is even pre-Indo-European, so the roots are not really well known. But anyway, they have very funny names like Gune, and Gune means a place where people gather to do something. So we would be in the CIC, the Center for Cooperative Research, on the place to do biomaterials, okay? And then there are the 3 CICs, Nano also in San Sebastian, Bio in Bilbao, and Energy in Vitoria. So when these centers were created, the idea of the Basque Country was that we would be in charge of promoting collaborative efforts within the region to create new industry sectors so that the industry would be diversified in the region. And so this is what we have been trying to do for the past 20 years or so. Currently in Biomagune, we are 12 PIs, and in total we have more than 100 researchers and more than 50 PhD students. We profit from the existence of the ICER-Basque Foundation, which also promoted by the Basque government, which is in charge of attracting scientists to the Basque Country. So they essentially invest in people, attracting good researchers with a quite healthy budget. And so we profit from that because this helps us give better conditions to the people who we bring in. And so thanks to that, not only via Malgré, actually there is a strong network of basic research and technology centers all around us, as well as some brand new companies. For example, Viral is doing gene therapy for cancer. There is also a health research center right next to the local hospital. So it's very easy to find collaborations and resources that are complementary to what we have. And so actually in the field of nanoscience, well, also because I'm an editor, we published this little piece in Nature to describe how to read the scientific environment. Anyway, so in Malgré, we have, as I said, a very small a very small group of people, only 12 PIs. And we have actually quite a healthy infrastructure, including rather well-built bioimaging facility, which combines in a very small space cyclotrons where we can generate isotopes. We have a small animal house for mice and birds. We have PET and CT imaging and 2 MRI machines, all in a very small area of, say, 200 square meters or so. And so this is in the basement, and then on the upper floors we have labs for developing materials for theranostics or for synthetic bioengineering. And so it's all very compact, and we have also a culture of joint work and sharing all the instruments. So this makes it a very attractive work to work. So within this environment, I started in 2012 the Bio-Nanoplasmonics Lab. I was always very proud of the name because it quotes 3 very important keywords for funding: bio, nano, and plasmonics. And so we were reasonably successful with funding, and we could create a group dealing with a number of topics, ranging from nanoparticle synthesis. This is something that we had been doing for 20 years before I moved. So we were already quite skilled on that. But then we were playing with surface chemistry, studying effects of plasmonic heating from a very basic perspective, but also with the potential application in mind. And then a lot of our research is based on surface enhanced RNA. I will tell you more about this during the lecture. And so my lecture today will focus mainly on this lower part here, related to surface enhanced Raman biosensing, bioimaging, and trying to contribute to understanding how biological systems and disease are working. Okay, so the main motivation for what I'm going to present you today is the connection between creating new cell models that can be more representative for studying cancer research. And so, as you know, in order to develop drugs, you have to go through plenty of phases so that your drug will be approved. Typically, from design of the drug to successful implementation, it takes at least 10 years. And only 5% of the drugs that are designed finally make it. So you need to have a lot of models, starting from cell models and then going to animal models into human volunteers, in order to develop this. So we wanted to focus mainly on the initial part, trying to create models that avoid the limitations of the standard 2D cell culture, where you have many environmental aspects which are very different to what happens in your body. Okay. And so we wanted to go into 3-dimensional models. The simple ones look like this, like just aggregates of cells. And still, if you want to understand how things work in such an aggregate, you need something that allows you to do biosensing so that you can monitor processes that are specific to this configuration of cells. And you also need to be careful about what is around the cells. So you need to take care, in the case of cancer, for example, you need to take care of the composition of the tumor core and the stroma around the tumor. So this is something that we actually had never done, but I'll tell you how we developed it. Okay. So our core expertise is in plasmonics. For those of you who are not familiar with the concept, It's actually not so difficult. It's related to materials that carry free electrons. So when an electromagnetic radiation comes and interacts with these materials, it creates oscillating charge waves. If you have a bulk material, at a certain frequency you can create this oscillating wave inside of a metal, for example, which is called the bulk plasma frequency. If you have a very thin film, then the oscillation is confined at the interface between the metal and what is next to it. If you have very tiny particles, there are oscillations that, in the extreme case of very small spherical particles, they will be dipoles. But then I'll show you later, depending on the morphology of the particles, you can have also other types of oscillations of electrons, which become very interesting for many conceptual advancements, but also for many applications. So in the case of biosensing, the traditional way of applying surface plasmon was the so-called SPR sensing, applying this concept, where there is a laser incident on a very thin metal film, which is coated with bioreceptors. So the idea is that there will be a specific angle for a specific wavelength of the laser at which this resonance condition will be fulfilled. And so this you can detect as a sharp peak. If the molecule that is specific of the bioreceptors on your surface is coming around and binding, there will be a small change in the refractive index right next to the surface, and this will slightly change the condition following this equation. So you will detect the absorption of the complementary of your bioreceptor. So this is commercially available, mainly used for basic studies, but you can also use it as a sensor. Now, in the early stages of plasmonics research, there was a lot of work related to LSPR sensing, applying the same concept to the changes in the oscillation of electrons in spherical particles, typically, when there are also absorption of molecules on the surface of the particle. So this has the advantage that the peaks are typically sharper than in the case of the SPR, and also that you can miniaturize. You can even apply a single nanoparticle as a sensor to detect the molecules. However, the implementation of these systems in complex biological matrices is not as simple as it looks. And so, even though there is still research connected to this, it's not so popular anymore. And there have been many other advancements related to this. Another of the typical LSPR biosensors, even if you didn't know, is the lateral flow tests. So here we have, you probably all know how it works. So you have a stripe, and then you dip the end of the stripe in the liquid where you want to do the test. And then there is flow, and on the flow, the liquid is carrying gold nanoparticles typically. You can also do it with other particles, but typically gold nanoparticles, which carry antibodies that are specific for the molecule that you want to detect. In the case of pregnancy tests, this would be the gonadotropin hormone. And so if the hormone is present, the particles that are flowing on the capillary stripe will also bind on what we call the test line, which will tell you if the test is positive or negative. And then we have a second line to make sure that the particles have made it far enough so that the test is valid or not valid. And this is exactly the same thing as we all have been using during the COVID pandemic. Okay, so this has been around already for 70 years or so. And so we have been applying nanotechnology for so long. I don't have the slides today, but actually the same concept of looking at colors has been applied for thousands of years. So the famous Lacour stuff is on display at the British Museum. Most of the Middle Ages cathedrals carry metal nanoparticles from the stained glasses. So anyway, we have been employing nanotechnology for ages, but now we understand a bit better how it works. All right. Now, one of the beauties of employing LSPR rather than SPR is that you can play around with the morphology of the nanoparticles. And by doing that, you can tune very finely the position of the resonance wavelength. So in this case, I'm only showing you what happens for those nanorods by simply elongating a little bit or increasing the ratio between the length and the width of the nanoparticles. You maintain one resonance, which is typically associated to oscillation of electrons across the axis of the rod. This remains more or less constant, but as you elongate the particle, this other resonance along the axis redshifts gradually, so you can pick exactly the wavelength at which you want to work for your application. And even more, for what I showed you earlier with LSPR sensing, the longer the particles are, the more sensitive they will be to changes in the refractive index. Okay? All right, I'm not going to expand a lot more on this. Let me just show you a library of particles that we have been making during the years. It's not only the beauty, I think they are beautiful. Actually, I didn't include it, maybe I should. Recently, there was a bit of a fuss that probably you have not seen, but anyway, there was a bit of a fuss because the fashion company Versace displayed what they call a scientific project on the social media, where they were actually describing the properties of nanogold and showing some of our pictures that they colored. So, you know, this all becomes, and they were sort of making a parallel between, for example, these arrays of gold cubes with some designs of the fashion that they make. Anyway, I'll show you later. So they are beautiful, and I think for my taste, they are beautiful because we managed to get very high monodispersity in each of them. And the monodispersity is very important. Remember what happened if you elongate a little bit the morphology of the particles, you get the shift. So you want to get well-defined resonances, you need to have the particles all look the same. Another important issue is that, as I mentioned earlier, depending on the morphology, you can have oscillation of electrons that lead to enhancement of electric fields at different positions on the particle and also different intensities, which will be very important for what I'm going to explain next. So, if you want to work with these particles for biological systems, you typically want to go into the so-called biological transparency windows. And so, because we can manipulate the plasma resonances with the morphology of the particles, we can use the morphologies and the sizes that we need for working in this window. So, in this window, there are several advantages. One is because the energy of the radiation is not so high, you're not damaging the cell so much. And second, because water does not absorb so much in this region, and there will be more penetration of light. So if you want to study either synthetic or natural tissue, you can go deeper in your samples. Okay. Now, because I was a bit disappointed with the LSPR sensing, some 15 years ago, I started working on this other concept, which is Raman scattering. So Raman scattering is very useful for biological studies because you can avoid the absorption by water of infrared light almost completely. So water is not very responsive to Raman. And so in Raman, what happens is that you're exciting electrons which are in the ground state, and then you excite them typically with visible light. It can also be near infrared. And then you take electrons to an excited, typically forbidden state. And then they relax back and they emit a photon, which can have a different energy to the incident photon, which is related to the vibrational frequencies of the molecule that you're exciting. And so you can look at the vibrational fingerprint of your molecules by looking at the difference in energy between incident and emitting photons. The problem is that because you're involving forbidden states, the probability of this process is extremely low. It's less than one photon in a million photons that get scattered in this way. And so if you want to apply this for real applications, especially where you need high sensitivity, this is very complicated. You will need very low accumulation times or very high intensity, which you don't want. So, almost exactly 50 years ago, Fleischmann and his group in Southampton discovered some special effects when you're studying Raman spectroscopy of molecules absorbed on metallic electrons. I don't want to discuss this very much. We all admit that Fleischmann's paper is the precursor of surface enhancement, even though if you look at the paper, you will not find any intensities for any enhancement. But anyway, following that, Van Dyne and others, also most of it's near here, were the ones who actually promoted the study of the plasmon influence on this process. So because of the 50-year anniversary, we are now having a series of perspective articles in ACS Nano that were introduced in this editorial. So if you're interested in learning more about different aspects of this technique, you can follow the different perspectives, and I'll give you another reference in a minute. Now, I showed you earlier these maps as well, where depending on the morphology of the particles, you can excite local electric fields, which can be localized very strongly in very small spaces. And depending on the morphology, they can have a lower or a higher intensity. The nice thing is that if you increase the local electric field, the Raman signal will be enhanced by 10,000 times the increase of the local electric field. So if you get electric field enhancement of say 10, you can already enhance your Raman signal by 10,000. If you get 100, I'm just guessing, you can increase by 10 to the 8th. So this has already been demonstrated many years ago that you can even detect a single molecule, because you can get enough enhancement to actually overcome the problem of the forbidden states. All right, another way of doing, so the problem is that the molecule has to be exactly here, okay? Another way of doing this is bringing particles close to each other, like it happens in most of these surfaces. And then the molecule has to be located on the gap between two particles, so that you can also see a large enhancement of the electric field. And this is what we call hotspots. I like to show these two movies to explain the hotspots. One is a simulation that we made of, well, it's actually a cartoon. Oops, is it not working? Yeah. Okay. Yeah, the cartoon is not working for some reason. Anyway, so here you will only see that the particles are coming together, and as they come together, there will be a large increase in the signal in the electric field. This is the real one. This is actually an experiment that was made also close to here, published in PNAS a couple of years ago. And it actually shows a parallel of the hotspots at the macro scale. So these are two grapes which are placed inside a microwave oven. And then because of the different scale of the object and the wavelength of light, you get hotspots right at the gap between the two grapes. Okay, so the effect is very similar. You are exciting something that gets concentrated at the gap, and then you get a large increase in the field. Okay. So this is the reference I wanted to give you. This is already becoming old, but still very popular. I collected, or I convinced, like 60 experts around the world to contribute on different aspects of surface enhancement, and we published this almost 100 pages long review article. But actually, I think gives you a very good overview of what has been done and what can be expected of this thing. Now, anyway, this is my own list of pros and cons of the surface enhancement, which mainly explain why this is not yet in the market. The pros, I already gave you a few. So apart from the high selectivity and sensitivity, you can get very fast experiments if you're lucky, if the conditions are good. You can use also portable instruments. I'll show you encoding. This is, I think, one of the most promising applications. And it can be applied almost to anything because most molecules have a Raman fingerprint. So you can in principle distinguish any kind of molecule. Problem is that substrates are not yet perfect. And I'll show you in a minute. Even if they are perfect, there will be other reasons why it is, I would say, impossible to quantify. Another problem is that if you work with complex systems, you can easily contaminate your signal because there will be many other molecules floating around. Another very important point is that the molecule has to come very close to the metal surface because the electric field enhancement is very, very local. Only a few nanometers away from the surface, there will be no enhancement. And I'll show you also an example of what happens if you want to use the same substrate over time. because you can get memory effects, so you still see at later times what was absorbed at the initial time. So all of this we have been trying to resolve over the past 10 years. All right, so let's see how this looks in a simulation. So nowadays we have access to either commercial packages or colleagues who can do extremely sophisticated electromagnetic simulations. So I give them a TM image, and they make a model that looks exactly the same, and they calculate the interaction of light with a certain wavelength, and they calculate where the Raman signal will be enhanced and by how much. So in this map, the white colors would mean a very large enhancement of the electric field, and the black ones would be no enhancement. And as you can see, very few spots are white, meaning that the signal that you get from an area of nanoparticles comes actually from a very small number of spots. Everything will screen what happens in other places. Even worse, depending on the concentration of your particles, you may have positive or negative effects. So, for example, in this is not displayed very well, but this is a collection of gold nanostars in a simulation, which are very efficient when they are isolated. But when they come very close to each other, you can even get destructive interference of the plasma resonance. So you get even lower enhancement in this area. So you have to design your system very well in order to obtain the enhancement that you need. So this is a summary of what happens if you increase density uniformly on a substrate for spheres, rods, and stars. And here you see for spheres and rods, you can always get an increase if you increase the density of stars in the same area or of nanoparticles. And if they get very close to each other, then you start to have hotspots. And this is why the slope here becomes higher. For the stars, you still get a higher intensity, but you don't gain anything by increasing the number of them. And this is actually something that we have seen experimentally as well. All right, so we need to design appropriate substrates. And this is something that we did already like 15 years ago, when we applied for our first European Research Council grant. These are actually grants that you want to have if you're in Europe, because they give you safety for 5 years, in this case for 6 years, because we applied for an extension. And you can propose things that allow you to do something far away from what you have been doing. So we proposed to apply surface enhanced Raman to listen to bacteria, to see how bacteria talk to each other. So this is a process that the microbiologists call quorum sensing. And it's a process by which bacteria release certain molecules, certain metabolites, that then other bacteria can recognize by surface proteins. And then when they do, they change the genetic cycle in the bacterium and they do something else. For example, they can decide to create a biofilm to start an infection, or maybe to run away because there's something that they don't like. And so I hired a microbiologist to do this, Gustavo Bodelon, and he suggested that we start by looking at Pseudomonas aeruginosa, a very nasty bacterium, which releases phenazines as communication molecules, which in some cases have very efficient Raman scattering signals. And so by studying these phenazines, you can almost forget about contamination. But still, we didn't want to have lots of nasty stuff in the bacterium medium to come on top of our substrates and block everything. So we actually decided to design very efficient and very clean substrates. And to do that, we first went to the simulations, and our colleagues told us, if you build something like this, you will obtain a collection of hotspots at every plane in between two layers of nanoparticles. But they have to be well ordered and of the right size. And so we struggled, but in the end, we managed to get something like this. So this is CM field tomography. So you see in 3D how the particles are organized in multi-layers. Perhaps not as perfect as these ones, but they seem to be working quite well. And apart from that, we wanted to screen the access of large biomolecules at the gaps or block them. And so what we did was to create an array of towers made of nanorods, and then infiltrate them with mesoporous silica, so that the small molecules, the phenosins, will infiltrate through the nanometer-sized pores of the mesoporous silica, come at the gaps between the nanorods multilayers, and then give the proper signal, while the larger molecules could not fit in between. So by doing that, we could incubate the bacteria directly on top of these protected nanorod supercrystals, and then monitor the increase in the concentration of the phenosine as they were proliferating and starting to communicate with each other. I'm not going to go very much more in detail of this. We did many more studies related, for example, to studying the interaction between two different bacterial strains, different messengers, so to say, and how, in some cases, one type of bacteria would actually destroy the communication of the other and win the battle, so to say. And so, we sort of started to understand a bit more of the behavior of these systems. Now, at the end of this project, we thought we had to try and get another of these good funding. And then we decided to first go for different substrates, try to design something that does not use so many nanoparticles and maybe is equally efficient. So we discussed with my colleague, Agustin Me in Barcelona, who could prepare templates where the size of the holes that we used to organize the nanoparticles were a few hundred nanometers, and the separation between them was also a few hundred nanometers. When you do that properly, you obtain arrays like this, which display very bright colors, different to what you would expect from the absorption of the nanoparticles, because here you're combining the plasmonic effects with light diffraction. So you can obtain what we call lattice plasmon resonances, and by doing that, you get very bright hotspots in between each of the nanoparticles that are comprising. So this is one of the dots in this picture, okay? So this is a very efficient one, very well organized. It really gets rid of the problems of uniformity and reproducibility. So this is what we decided to propose how to use in the initial stages. But we wanted to go much farther. We wanted to propose something to understand a bit better how cancer works. So I don't have to explain why this is important. The concept was we first are going to design these very efficient substrates to study processes that are specific to cancer. And we are going to create three-dimensional systems, three-dimensional cell culture that look better like a real tumor, and where we can label the cells with what we call cell stacks, which are labels that depend on the Raman fingerprint of molecules that we specifically absorb in the nanoparticles. And so by doing that, we proposed all of these challenges. This was 2018. We proposed real-time detection of metabolic processes specific to cancer, fabrication of 3D tumor models using scaffolds, incorporating nanosensors with all tumor-relevant cell lines and biocompatible so that we could also study over time. And then monitoring real-time the labeling of different cell types and using circumfocal microscopy, meaning that we can do all this monitoring in 3D over time. And so finally, we wanted to also apply this as a drug testing system. So this was what we proposed. They bought it, and we got the funding. And so we started with this schematic view of what we wanted to do. Okay. So as I mentioned, we want to do biosensing mainly related to the alteration of metabolic processes in tumors. We wanted to create scaffolds where we could embed our nanoparticles as sensors, and we wanted to use the labels so that we could know exactly what type of cell we are looking at in each position in 3D. Okay. So we don't know about everything, and so we decided to team up with a group of Arkaitz Cartel at our sister institute, CC Biobuni in Bilbao. Arkaitz is an expert in cancer metabolism, and he proposed a few types of disruptive metabolism reactions that we could look at. And the simplest one that we found was the conversion of tryptophan to uridine, which is catalyzed by the enzyme IDO1, which is actually overexpressed in many cancer types. And so by using these organized substrates, my student Javier Clo, who is now a postdoc already, he started looking at the Raman signal of the two metabolites so that we could characterize them properly, finding peaks that are very specific to the presence of these molecules, and then simply incubating, in this case I think it was simply HeLa cells, taking supernatant and then dropping on top of a substrate. He was watching over time the decrease in the intensity of the peak characteristic of tryptophan and the increase in the intensity of the peak of uridine. So this was our first example of how we could try to monitor these processes over time. Now, a second process that was very interesting to us was the influence of cell death, mainly because the type of cell death and the cell death mechanism can also affect the behavior of tumors. And actually, there were many literature examples where you can find that the release of metabolites after cell death can be anti-inflammatory but also pro-inflammatory, which affects also the development of the tumors. And so Javier started looking at what happens if you induce cell death on cancer cells and what type of metabolites are being released. And so during this process, he could also monitor this conversion of tryptophan to uridine, but there was, if you see, another peak that we didn't see before that was getting increasing intensity. And this we could assign to purine derivatives, which are related to cell death. And in fact, we were promoting cell death by different mechanisms and consistently seeing this peak that was not present in the control of the healthy cells. Okay, and now you will tell me very rightly that we are looking at cell supernatants where there are lots of stuff and we are just extracting information from a single peak. And I said, you're completely right. On top of that, we are looking at this process over time. And I'll show you this example here by using one of our substrates, where we first incubate with, I think this is mercaptobenzoic acid, and we see this very strong peak. And then we clean it, and we immerse it in nicotinamide, where we would expect a strong peak here, and we don't see it, we still see the peak of MDA. And the same if we incubate with a mixture of both. The reason is that MDA, which contains a thiol group, absorbs very strongly on the metal, and meaning if we wash, it doesn't go away. And this is what I call earlier memory effects. So in fact, in the early stages, we had to do something like this in order to monitor over time, place one substrate, measure directly on top of the glass, and then remove it, and then come with another and put it on top of the sample and measure again. So we had to renew the substrate every time, potentially also disrupting the sample we are measuring. And so my students, I don't want to claim any novelty or ideas coming from my side, they decided to employ another effect of the plasmonic system, which is the release of heat. So when you're exciting with a higher intensity laser, apart from increasing the intensity of the local electric field, part of the radiation is absorbed by the metal and released as heat. So if you cover the metallic substrate with a thermosensitive polymer like TLGA, and then you come with your laser at a slightly higher intensity, you can actually burn a hole on the polymer, and then you can do a measurement directly on the nanoparticles that are exposed, while the rest of the substrate remains protected. And then when you do that, you can do a measurement in one place, clean, incubate on a different substance, drill another hole, measure again, and so on and so forth. And the same thing you can do for the cell culture experiment. So here you can do the measurements in situ and without changing the substrate, and by many times. We calculated that for the area that we are using here, we could do like 10,000 measurements or something. So anyway, this was an improvement that allowed us to go much faster. And this is when we started to think about high throughput analysis. And so we started to do hundreds and thousands of measurements so that we could implement more sophisticated data analysis, not just rely on one peak to detect one molecule, but actually studying the full spectrum, and then, for example, by principal component analysis, looking at the separation of components over time for different cell death mechanisms. And so, for example, if you're comparing the control with the effect of UV light, UV is known to affect the DNA in the molecule, in the cell. And so death is slower than, for example, if you heat the cells. And so the signals for the control and UV were actually very close to each other at early times, but they got gradually separated at later times because some molecules were being released as the cells were dying, while the control was still healthy. And then we even implemented also machine learning techniques, so we could actually train the algorithm to distinguish initially between dead and live cells. And later on, we could distinguish, for example, between apoptosis and necrosis. And here we can apply already to our known systems. And then by applying this algorithm, we can decide whether we have one or the other cell death mechanism. In fact, when we were studying this, I think I'm going a little too slow, so, but I'm trying to make you understand. And so I'm going to describe this very quickly, but by learning different components within the spectra of different cell death mechanisms, we thought that maybe we can try to discover new things by doing this high throughput screening of samples coming from cancer cells. And so again, in collaboration with archives, we started to look at the MTA, the methionine salvage pathway, which is related to some process that takes place in many cancer cell types, but not in all of them, where in some cases this MATP enzyme, which deletes the release of MTA from the cells, is actually this enzyme is deleted in the characteristic gene of certain cancer types, and then this prevents the recycling and the generation of pure impulse, which are important for the development of the tumor. And so when this happens, the tumors are more sensitive to certain drugs. Now, by doing studies related to the monitoring of extracellular MTA, plenty of control experiments, and then comparison with the standard qPCR and other biochemical tools, we actually discovered that in some cases where macrophages are also in the neighborhood of the cancer cells together with fibroblasts, there is communication between the cancer cells and the macrophages, so that the genetic evolution of the macrophages is changed. So the macrophages are used to regenerate the pure impulse so that the tumor can actually continue developing. So they found their own way to escape the deficiency in the production of MATP by talking to the macrophages and making them release what they need. So this is something that was not known before, and maybe will help also generate other studies in related things. Okay, but all of this is still done in the standard 2D culture. Okay. And as I mentioned, we want to go into something that resembles more faithfully how a tumor looks like. And so we want to escape from here, but we don't want to come here because it's too complicated and we don't find enough volunteers for that. So we decided to start doing 3D bioprinting. We had to admit we knew nothing about that at the beginning. But since we got the funding, we bought a very nice 3D bioprinting. And I was lucky to come also with a polymer chemist in the lab who was extremely good at designing the initial inks that we need for that. So the idea was, again, as I mentioned earlier, to 3D print the scaffold containing nanoparticles as sensors, and then to incubate with cells of the composition that we need so that we can recreate a tumor in 3D. And so initially, as I say, we had to develop an ink that we could print with the morphology that we want. which remains in shape after printing. So rheology and curing was all being developed by my students and postdocs. And within the hydrogels that are composing this 3D scaffold, we have gold nanorods. Okay? And because these hydrogels are porous, we expect that molecules released by cells will infiltrate and get close to the nanorods, so we can do the surface enhancement detection. So this is what you can see in these plots. For example, we incubate with adenosine. We have a very characteristic peak, and we can do confocal surface enhanced Raman imaging of the scaffold. The green color means that we are seeing this spectrum. And so this image is telling us, first, we can faithfully detect the presence of the molecule. Second, we can detect it everywhere because the distribution of nanorods is very efficient, very uniform. The areas that look black, in principle, my interpretation is that either the molecule could not diffuse enough or there was not enough content of nanorods. But I think that the image shows that we can have a faithful reconstruction of the shape that we have. Now, the second part is labeling the cells with codes that will allow us to distinguish the different cell types and that do not have the problems of photo bleaching or using different lasers for excitement. And so this is what we call cell stacks. So we make nanoparticles. We code them with what we call Raman reporters, which are molecules that attach strongly to the nanoparticles and have a very well-defined Raman fingerprint. Then we protect them with an extra layer that will avoid leaching of the molecules and also will avoid contamination of the signal by other molecules coming from outside. And this external layer we can also label with antibodies or other recognition molecules if we like. And so an example looks like this. We have a gold nanostar. We coat it with thiolated aromatic molecules that have a very well-defined Raman fingerprint. We coat them externally by an amphiphilic molecule that forms a rigid layer, which becomes again hydrophilic, so it can go into the cell culture medium. And then we incubate with cells. The cells eat up the particles which are encoded. And then we can monitor by Raman microscopy the presence of particles inside of the cells. And this we can do for a large library of Raman reporters. Each of them will have a different fingerprint. And we can use either nanorods, nanostars, or other shapes as the Raman reporters. So electron microscopy is telling us that, as expected, the particles get internalized by endocytosis. And if we now incubate different cell types with cell stacks with different codes, and we co-incubate, we can actually monitor the behavior of these cells for a long time, and clearly distinguishing each of the cell types from the color coming from each of these codes. As I say, this is done by using the same laser beam, typically 785, which is very benign for the cells, and there is no photo bleaching of the signal that we are measuring. Now, again, this was still 2D. We showed already a few years ago that we can do this in 3D by simply depositing two layers of cells on top of each other. You can see roughly how they sort of interdigitate. And so this shows that we are in a good position to start thinking about using the scaffolds as the templates to grow the cells in 3D. And so this is something that we also managed to do after some trouble. And then again, we can do simultaneously imaging, in this case, so again, of this adenosine. The green color means that we are detecting the metabolite. The red color comes from the cells that have been absorbed on the 3D scaffold. So here we can do this experiment in 3D simultaneously with the same laser beam. More recently, we published this study where, by again using similar scaffolds, we can infiltrate the gaps with, for example, matrigel, which allows you to grow cells in 3D. And then over time, we were watching the aggregation of cancer cells together with cancer-associated fibroblasts and starting to form the initial spheroids that then give rise to the formation of tumors. So this is something that we could monitor initially also by fluorescence microscopy, but then with cells we could also monitor for longer times. In fact, this is an example for breast cancer cells, but you can use it actually for many different types. Anyway, the initial example with a similar system for a drug screening system is printing the scaffold in this sort of microfluidic chip with a reservoir where we can inject an anti-cancer drug. And then we can infiltrate the holes in the scaffold with matrix gel and with cells that express the new fluorescent protein. So we can also decide whether the cells are alive or dead with fluorescence. And then we can inject a drug here, and we can monitor over time the diffusion of the drug by sensing on the scaffold. This is what you see here, 1 hour, 2 hours, 24 hours. It takes a while for the drug to diffuse through the whole system. And then we can compare this diffusion with the changes in the distribution between green and red, which means live or dead cells. So this is already a system where we can study the effect of drugs over time with cells distributed in 3D. Now, in a parallel sub-project, we wanted to print models that looked much more like the real system. So if we go to the literature, we find that both for mice and for humans, if you look at breast cancer, you typically have a much denser core made of cancer cells, and then a stroma which contains fibroblasts, epithelial cells, collagen, and a few other things. And so we wanted to print this. In order to do that, avoiding the use of, say, artificial extracellular matrices, we decided to go the hard way and employ extracellular matrices that are derived from animals that look close to humans. So in collaboration with this Biodiagnostic Institute, which is next to the local hospital, we started to use their corpse from surgeon experimentation, and then collect tissue from the breast, and then extract all the components, the cellular and DNA components of the animal, and then create an ink on which we can culture human cells. From this confocal microscopy image, you can already see that they spread very well. They feel comfortable in these matrices. And then we could implement a dual bioprinting process by which we could print the tumoral core within a stroma that looks like the one that we have in the animal. And so here in green would be, in this case, NCF7 breast cancer cells. And then here in blue and pink, you will have the epithelial cells and the fibroblasts. And this is all embedded in this decellularized extracellular matrix from the animal. With this system, we started to do also tests of the effect of different anti-cancer drugs. And then we tested also differences between different types of breast cancer cells, NCF7 compared to triple negative cancer cells, and then employing drugs that are known to be more effective for one and less effective for the other. And with these 3D models, we find very similar results to what has been reported for humans. More recent work was related to employing again the 3D system, the 3D confocal imaging, both with fluorescent and with SIRS. And again, depending on the time that we are doing the observation, we can compare the stability of the core, meaning that we are looking at the possibility of emerging metastasis. So here we have NCF7 cells in the core. Here we have triple negative. Here we have triple negative cancer stem cells, and we are imaging in this case between day 1 and day 5. For MCS7, the core remains more or less stable. For triple negative, we start to see the emergence of certain cells that are escaping the core, and this is even more evident for the cancer stem cells. So we are able to propose this system also to watch what happens at longer times, and maybe to start predicting what happens in the formation of metastasis. I'm aware that this is a very simple system because we have no vasculature, for example. We have some results related to that, but they are too preliminary, so I prefer not to present them today. But there are actually many very interesting developments that you can connect to this system that allow you to go even further in the reliability of these models. I wanted to show you that we have a similar system for melanoma. So again, by using the DECMs from ticks, we could extract the different layers from the epidermis, dermis, and the basal membrane of the skin, reproduce the 3-layer system for incubating human cells, and then also implementing melanoma cells on the epidermis, and watching over time how some of them we are migrating through the basal membrane in a potential start of metastasis. So with this, I think I made a broad summary of what we have been doing in the past 5-6 years in our efforts to help transition between current medicine and future medicine, where you will be able to perhaps classify patients according to measurements that may not be so exhausting and so invasive. And so maybe in the future, apart from the physical exam and PCR test, we can do a search test to decide whether one patient should be treated with one drug or with none. So I'd like to finish by thanking many people in the lab. This is the group in San Sebastian currently. Javier and Pablo were not in the picture, so I added them because of the relevance in the initial work on the detection of the tumor metabolism studies. And these are the people in my second lab in Vigo. They are not connected to this work, but they are starting to do also very nice research more related to other fields like chiral nanomaterials and things like that. And my last slide is dedicated to announcing that we are organizing the next Gold Conference in San Sebastian May next year. So please feel invited. It will be a very lively scientific environment and hopefully also an entertaining time outside of the lectures. Thank you very much.
Thank you so much for this really exciting talk. Great collection of work on different topics that really covered many of our interests. So I'll open the talk to questions.
Did I understand correctly, in the 3D cell model, you were doing both fluorescence and cells? So in the cells, how you were using individual nanoparticles, I guess. Because I don't see how you could build your array inside the cell.
We are not doing fluorescence and cells simultaneously.
Oh, no, okay, but for the cells, how did you?
For the cell, what we do is we use these cell stacks that are internalized in the cells.
Yeah.
So we are looking at signals from cells that contain those nanoparticles.
But where is the nanoparticle?
Inside of the cell.
So that's single.
Exactly.
Okay.
Yes.
And do you know anything about the percentage of cells that are pregnant?
No, that's a good point. That's a good point. We actually have done many more studies.
Yes.
Where we even look at the possibility of cells releasing the cell stacks by exocytosis. And our results tell us that this is not a very efficient process. So we design systems where we could monitor a certain number of cells. The efficiency seems to be quite high. It's difficult to tell how many, but those that we can image for a long time without really losing the signal. Now we are doing a bit less realistic, but more sophisticated studies where we encapsulate a few cells labeled with different types in microfluidic droplets, and then watching over time whether there is cross communication between them. And so in some cases we do see some, so we have to be careful, as I mentioned, in very long studies. It may be important to realize that the signals may be changing. What we could also do is perhaps reducing the initial fluorescence studies, and then in that way we are not photo bleaching anything at the beginning, and then maybe at a later stage we can do another analysis to sort of correlate with the cells image.
I was thinking more if you're looking at a living cell culture that's proliferating, the new cells.
Yes, we also have been looking at cell division. It looks like the daughter cells are carrying also nanoparticles, so there's a enough number that they split. It's difficult to tell whether all of the daughter cells will carry
Okay, so you
nanoparticles.
have a large ratio of nanoparticles to cells.
Exactly. So this is of course a very brief summary of also work by many people. So we actually studied the loading that allows us to have enough signal that can replicate without being toxic for the cells, which is always tricky. But we try to be systematic and understand what we're doing.
Are they stars or rods?
We use both. Yeah.
Thank you for the nice presentation. So I will have two questions basically. The first one would be for the transition to the medical application. I gather that you want to basically overcome the limitation of the actual cell culture by using basically the cells of the patient and growing then the tumor that represented the artificial tumor. So how long does it take actually to grow the tumor itself with your technology?
No, that's not very complicated. The complicated part is obtaining the cells from the patient.
Yeah.
So this is something that at the moment we are struggling with. We are working with samples that we obtained from the National Center for Oncology Research in Malgré, because the local hospital is still reluctant. So we are working on an ethical protocol that will allow us hopefully to get samples from local patients in the future. And so now we work with collaborators. Expressing and expanding these patient cells is also quite laborious. So this is the ideal case. In fact, if you want to work directly with patients to convince the surgeon to diagnose a certain drug, I think that this is very wishful thinking, and this is what the director of the oncology hospital told us. So our objective is not so ambitious, although we would like to do that. Our objective is more teaming up with pharma companies so that they reduce animal experimentation and use these models to do more advanced testing of drug candidates. And we are working now with automated prepared sausages so that we can actually do high throughput production of these methods and then high throughput testing of drug candidates.
Yeah, also because implanting a tumor in an animal model is not
exactly.
Tried and
Exactly.
Actually some other drugs.
Something we are also trying, but this is also very complicated because of the heterogeneity among patients. We are obtaining also breast samples from breast reduction surgery from women. And we are creating the cellularized CCMs for that. But the heterogeneity between patients is enormous, depending on age, weight, and a few other parameters. So it's very difficult to have a consistent protocol that will allow us to create a mean that is reproducible enough.
And maybe the second one will be short. You mentioned that you actually check for necrosis and apoptosis as the two mechanisms for cellular death. Some protocols, some drugs and some treatment actually induce also more senile cells that do not actually reproduce anymore. Is that something that you see value in checking also?
No, we haven't looked at that, but the main purpose of these studies was to show that this technique can be useful. And so what we are hoping is that more and more people will start, taking the risk of actually looking for something specific that will help understand processes like sensitivity of cells and so
Yeah,
on.
because actually it takes pretty long time to do the Western blot test,
Exactly.
so that could be.
So if you know well what you're looking for, and then you can establish a training protocol for the machine learning algorithm, then probably you will be able to do also a meaningful study.
Probably it's related to the earlier discussion. Are you interested in like integrating like the 3D scaffolds in organ-on-a-chip platforms in order to mimic more physiological conditions?
We are using the scaffold mainly to look at early stages of tumor formation, as I showed here. But we are also working on a different approach. So going from this core stroma model, we are actually printing sensors directly on these systems. So we can use the bioprinter to also print like rods or cylinders containing nanoparticles at the spot where we like. So we can print in the core, we can print far away from the core, and then we can do sensing at the different spots and then decide what is happening at different parts of the tumor over time. So this is an approach that we thought would avoid having so much stuff around the particles over time.
And for the ink, trying to use, for instance, materials like sensitive materials to mimic like mechanical simulations and stuff.
Yes, of course, mechanobiology is also very important in cancer. We try to implement different types of sensors, and in some we were not successful enough. We did not yet implement mechanosensors. We are actually together with a former postdoc in the lab who is now a junior PI. We are looking into using piezoelectric nanomaterials to try to look into that. We did look into nanothermometers, which were also printed in the scaffold. And we managed to get something which was responsive to infrared light, so looking at near infrared photoluminescence to decide on temperature depending on the ratio between two emission peaks. And it was working in clean samples, but as soon as you have a very complicated sample, the sensitivity drops a bit, and we didn't think it was reliable enough.
There is a graph that you showed that you had the Raman signals and the cell signals for the same molecule, and I will not try to pronounce the molecule, I'll get it totally wrong.
Yeah.
There is a broadening between the Raman peak and the source peak. Was that just instrumentation, or was there something?
No, there can be broadening, there can be even shifts in the position of the peaks. Because of several reasons, but one of the main reasons is that the molecules now are absorbed from the substrate. And so the orientation may be different, and the vibrations may be also be different.
We have a question from online. Yeah, one last question related to the last experiments with metastatic potential. Is there a possibility to get evidence of the signals that go out of the cell by exosomes or extracellular vesicles? Because this is a mechanism that is a bit advocated, or well, there's a controversy related to the actual role of exosomes.
Yeah.
But what is your take?
That's a good point. We also did some experiments having nanorods in the DCM. It's still tricky to do a detection which is clean enough. So if what you want to detect is a particularly strong Raman signal by itself, then there is a chance that you could see it. But you have to think that the density of particles in the DCM is not very high, because otherwise everything would be black. And so you also have to be lucky that what you want to see is diffusing all the way and flying through the particle.
So we end up with not a wishful thinking,
Exactly.
but observation. Okay. So thank you very much. That was
Thank
a really nice talk, excellent results, and